It's a real honor to have been chosen as the 2015 Peter Gallagher Memorial Lecturer, and it really is a pleasure to be here. I've had a chimeric career as a researcher and administrator, and so I thought I'd tell you a little bit about that journey and then end with some philosophical notes on what I think the future holds for the next generation. And I've got a particular agenda which I'm promoting with the next generation. You may agree or disagree. I had the good fortune of doing a postdoctoral fellowship with Sir Derek Barton, Nobel Prize winner in 1969, who used to tell me constantly that invention comes about under one of three scenarios: conception, misconception, or accident. And it's recognizing invention in each of these three scenarios that matters. And that's why we instruct our students in the art of creative and critical thought, so that they're able to identify and seize upon the inventions when they do occur. And so what I like to do at the beginning of this lecture is to use my own career with three vignettes to illustrate the concepts of conception, misconception, and accident. So let me begin with conception. Nature has given us many examples of natural products. And as you know, the Nobel Prize in Medicine was awarded this year to those exploiting natural products chemistry. In your own country, you have this plant which yields a compound. It's not important what the structure is. It belongs to a class called the alkaloids. And this particular compound inhibits certain enzymes. So it blocks the action of certain enzymes. It didn't take much for us to conceive of another class of molecules in which you notice that this nitrogen atom is replaced with the sulfur atom bearing a positive charge. We reasoned that this nitrogen would bear a hydrogen, also giving it a positive charge. And so it wasn't rocket science to ask the question whether the presence of a positive, permanently positively charged species would also act as an inhibitor of the enzymes. It turns out that that molecule you see there was an effective inhibitor of the same class of enzymes, and we went on to litter the literature with many, many examples of positively charged sulfonium ions. Right? These belong to a class known as sulfoniamides, which also exhibited some very interesting properties. Serendipity then entered my life because at about the same time, as soon as we'd completed that work, Yoshikawa et al. In Japan isolated from the roots of this plant, Salacia reticulata, known in Sinhalese as Kotara himbutu, compounds, and I'll show you the structures very soon, that also corresponded to sulfonium ions. Now this is an interesting story, and it is connected to serendipity because I grew up in Sri Lanka. My origin is Goan. I'm of Portuguese origin from Goa, but I grew up in Sri Lanka. And that's my maternal grandfather who suffered from type 2 diabetes. And he used to drink water out of this mug carved out of the roots of this plant as a cure for type 2 diabetes. And so this took me back to my roots, literally. Right? And so it was interesting, as soon as I saw these structures, and salacinol was the first of this class of molecules isolated by Yoshikawa, I realized that now nature was giving us what we had designed by analogy. And so we went on to synthesize many different variants, you see here, that was subsequently isolated from the roots and leaves of that plant. We were the first to synthesize this molecule in efficient fashion, and again, it's not important. The details are not important. Just suffice it to say that we were able to arrive at the natural product containing sulfur, but then being perverse, we decided, well, if now nature is giving us the sulfur compounds, let's make the compounds with nitrogen, and moving on then to selenium. These compounds are named after my graduate students. Ahmed Ghavami synthesized gavamiol, and Blintol is named after Blair Johnston and Pinto. We were going to call it Bleriol, but that didn't seem right. So Blintol comes from Blair and Pinto. Right? And all of these had very interesting properties. And they were inhibitors of the enzymes. We went on to synthesize a very large array of compounds, over 200 variants, and all of these had very interesting properties. And I'll elaborate on that in my lecture tomorrow. But the interesting question, of course, was, would they be effective in controlling blood glucose levels? And here are studies in rats where we dose them with a carbohydrate load, starch, and relative to control, you see that we're able to control effectively the plasma glucose levels if we administer salacinol or Blintol prior to the carbohydrate load. This acarbose is a molecule marketed by Bayer for the treatment of type 2 diabetes. So it appears that the rationale certainly is valid. Nature is now giving us sulfonium ions. By conception then, we had conceived of this class of molecules before they were isolated from nature. Let me just tell you about this very interesting class of molecules, or candidate of the class of molecules, Catalano. Tentative structure first reported in 1998, but you'll see for the chemists in the audience that the particular three-dimensional orientation about these centers has not been specified. And it was a race to deduce the structure of this molecule. And I found out later who I was racing with, and I'll tell you that in a minute. But just for the aficionados in the audience, 6 undefined stereogenic centers leading to 64 possible structures. We certainly were not going to synthesize all of them. We were going to use our heads. And so we had synthesized other analogs with 5 and 6 carbon chain extensions. Here you see a 7 carbon chain extension. And using our structure activity studies, we were able to narrow these possibilities down to 4 possible stereostructures. This turned out to be critical because others were busily synthesizing the different structures. And using one of the gut enzymes as a candidate, we were able very quickly to narrow the structures down to 4. And eventually, we won the race, and we deduced the exact structure of cotalenol. You see the wedges and the dashes indicating three-dimensional perspective. And I found out later who I was competing with. We're competing with 2 major teams in Japan, one led by Fujifilms and the other led by Mitsubishi. And so it was quite an event, and to their credit, they flew me out first class to pay tribute to our efforts. This was an interesting story because it took me back to my roots once again, this time my academic roots. This was my PhD supervisor, J.K.N. Jones, very famous carbohydrate chemist who brought carbohydrate chemistry from Bristol in the UK to Canada. And he, many years earlier, had isolated from avocados a compound called persitol. And this particular blue fragment is persitol. And so it took me right back to my academic roots, and we were able to synthesize this molecule a different way, starting from persitol. But with 2 very intelligent coworkers, we were able to beat teams of about 25 in each team. So the lesson here is that use your head, right? Think a little, and perhaps you'll be a winner, right? The real question here, of course, is can we use these compounds in control of better health and in control of disease? As you know, early humans were much more active than we, right? And excess of carbohydrates when available, were consumed. They were stored in the form of fats, and this was very important during times of low food availability. With today's lifestyle and changes in lifestyle, we generally ingest large quantities of carbohydrate in very short periods of time. We lead a more sedentary lifestyle, and so the energy intake exceeds the requirement. We don't burn as much glucose, right? And this has led to certain energy metabolism diseases, obesity, and type 2 diabetes. Right? This is a problem the world over. In addition, in infants, there are some carbohydrate malabsorption diseases, and I'll say a little bit more about this. This is congenital sucrase isomaltase deficiency. Infants missing this enzyme, living with chronic diarrhea from birth till very late into their teenage years. But we now recognize what it is. Okay? And part of that story is because of the compounds that we have discovered. So, starch, right? We consume a great deal of starch. And molecularly, it's a seemingly simple molecule shown here with glucose units attached to one another. It's not really important what these are, but it's a very complex architecture shown here. Okay? And certain enzymes break this starch down. So as soon as starch hits your tongue, you have enzymes called salivary alpha amylase that break those large fragments down into smaller units. Pancreatic alpha amylase then kicks in, keeps breaking them down. Then they go into your small intestine, and you have these other enzymes. I've mentioned some of them: sucrase isomaltase, maltase glucoamylase. These are in your gut. They're lining your intestinal wall. They break down these smaller fragments into glucose, and glucose is then absorbed through the intestinal wall into your bloodstream. When this goes wrong, you get increased levels of buildup of glucose in your bloodstream if the glucose is not shunted into your muscles, right? In type 2 diabetics, that is an issue. You have generally normal insulin production. In type 1 diabetics, the insulin pump is deficient, and so the shunting of glucose from the blood to the muscles is compromised. Right? So it's all about control of blood glucose levels here. This is not important for this particular lecture, but there are several forms. We have mapped all of those using our compounds, and we know now what each of these subunits targets. So I'm showing here different linkages, a 1,4 linkage, 1,6 linkage. Again, it's not important. This is sucrose, and that is broken down by one of the fragments. This is sucrase isomaltase as an enzyme, and we have teased all these activities out. So, we've characterized your gut enzymes, right? Can this help us? Can this help us in controlling the release of glucose, right? So there were a lot of questions we asked. You may have wondered about the origin of the phrase "Comment allez-vous?" That came about in the Middle Ages in France, and it was exactly as you think, right? It says, "How are you going?" And you know what they were referring to, of course. And that was gentrified, "How do you do?" Right? And I don't know what you say here, "How are you going, mate?" Right? But it referred to gut health precisely, and that was the origin of the phrase. And so this turned out to be a very important diagnostic. So we asked some very, very detailed questions. Why do you have so many enzymes? We asked, can we regulate those individual activities, right? Can we control the fast-acting enzymes to place the onus on digestion on slower-acting enzymes? Why do we want to do that? Because we want to modulate starch digestion. We don't want to turn it off, but you want to have controlled release of glucose. You don't want a spike of glucose. You want to extend it throughout your small intestine. You have about 10 feet of small intestine. And if the burst is at the front end in the duodenum, that's no good. You want it released continually right down to the ileum. And we found subsequently that if you have glucose deposited at the ileum, you can control gastric emptying of your stomach, and you can control satiety. You can control appetite, and so you don't feel the need to eat as much. And so this is a form of dietary control. So having a knowledge of these individual enzymes can moderate glucose release, we maintain. And these are very detailed questions about the gut microbiome. Each of us has a different supply of bacteria in our guts that have a redundant mechanism to the human enzymes. So if your human enzymes don't take care of it, the gut enzymes do. Okay? This shows a palette of many compounds that we have synthesized, and I can tell you that the bottom line is we are now in a position to modulate the different subunits of starch digestion. We can turn them on, some of the enzymes, we can turn them off. Okay? And so that's very powerful. And the questions we ask then, can we use these as therapeutic agents for the treatment of digestion disorders, and can we use modified starches therapeutically? So these are two strategies for controlling starch digestion. Our animal model is shown here. These are shrews, as in the taming of the shrew. Ugly animals, and they smell a lot, so I had to take off my clothes on the porch before I entered the house. I think it was apparently because I smell, but I think my wife liked it, but that's another story, right? So these are shrews. These shrews come from Nagasaki. So their ancestors suffered through those disastrous bombings. And interestingly, of those 4 enzymes that I showed you, these shrews are missing 1. So it makes it a very useful animal model. But the more important question, what about humans? And so this is one of our patients, and we asked the question, how can we treat, actually, this, I was there on his 2nd birthday, chronic diarrhea from birth. And this is tragic when you see this. And so we asked certain questions, can we reverse this sucrase isomaltase deficiency in this patient? And we asked the question, in the shorter term, could we supply the missing enzyme? Because we identified in these patients the absence of one enzyme. It was the same enzyme missing in the shrews. And so now we've just completed a clinical trial where we've fed the missing enzyme to children suffering from CSID. All 3 of these kids, siblings, were suffering from CSID. So that's conception. It all worked out. Boring. When it works out, it's boring. Let me move on quickly to misconception. Right? This is a diagram of the cell wall of a bacterium, Streptococcus group A. You see protruding from the surface different types of molecules. The long red chains are proteins, and you've heard of proteins. I've talked about carbohydrates. I've talked about alkaloids. You've heard about steroids, corticosteroids. This is just a different class of molecule. The orange chains are carbohydrates. Right? And so we were interested in designing a vaccine for Group A Streptococcus, as some of you in this institute are. It turned out that patients who were suffering from strep infections, if you took from their sera and isolated antibodies, that they bound to those carbohydrate structures that I showed you on the previous slide. So these chains. Furthermore, so we said, we'll use the carbohydrates to generate a vaccine, because the carbohydrates were common to all serogroups and types of Strep A, whereas the proteins changed markedly. There were 80 different serotypes. Okay? But there was a catch, because in the literature there were 2 reports that the antibodies that targeted the carbohydrate also interacted with structures on heart valves, right, and on heart tissue. So glycoproteins on heart were bound by the same antibodies that bound the carbohydrate. This seemed very strange, because I knew that the carbohydrates on the bacterium did not correspond to mammalian carbohydrates. Okay? Furthermore, immunization of bunnies with that carbohydrate led to circulating antibodies, which were claimed to cause lesions in heart tissue. Okay? So there were two reports in the literature, and I saw this, and being a young buck, I decided, well, you know, I think I know what's happening here. In the same way that this lava mimics this tree branch, we have a situation of mimicry here, where the carbohydrate is mimicking glycoprotein structures on heart valves. And I propose that there was a cross-reaction between carbohydrate structures here and protein structures on the heart valve tissue. There was a correlation in humans between strep infections and rheumatic heart valve disease. And so we set off on this great excursion to screen these libraries, you know, hundreds of thousands of sequences of peptides, these are smaller fragments of proteins, with our antibodies directed against the carbohydrates. And I was fortunate with several different bacteria. Let's look at Strep A. This is a very long polymer, carbohydrate in nature. We're able to simulate that with a 6-mer, a hexapeptide, different class of molecule. Okay? We looked at the shapes, and here I show the shape. This particular shape is very important to be recognized by the immune system. We went on to other bacteria. This is the group B, strep capsular polysaccharide. That we could mimic with this peptide. Again, this shape was important. This turn turned out to be very important for recognition by the immune system. With Shigella flexneri Y, again, we could replace this entire polysaccharide with an octopeptide shown here. Okay? We went on to crystallize the carbohydrate and the peptide mimic in the antibody site, and we were able to speculate about the nature of the mimicry. Okay? And we came up with a theory of functional versus structural peptide carbohydrate mimicry. Okay? And we postulated how these structures mimicked each other with respect to antibody recognition. Immunologically, they worked. We immunized with the peptide conjugated to a protein, and we got binding back to the carbohydrate or the bacteria. So we completed the loop. So we had a situation of carbohydrate peptide mimicry, and that was quite significant at the time because going between groups of molecules was fairly unusual. We worked with patients at the Princess Margaret Hospital in the Bahamas. These boys are suffering from rheumatic heart valve disease, and they would have to get injections of penicillin intramuscularly for the rest of their lives and never play sports. And so using these patients, we were able to withdraw sera and map their sera with some of the molecules we'd made. Again, this is not important, but to those of you who understand, this shows that certain carbohydrates bind to the antibodies in their sera. But the question that we had to answer, and that led us down this path, was, are those carbohydrate structures present on heart tissue? And so we worked with pathologists examining heart tissue from patients who had, some of whom had died from complications from strep infections, rheumatic heart valve disease, etc. And the red bits indicate carbohydrates on heart tissue. But this is only the control, where I've spiked the heart tissue with the polysaccharides. All our examination of all heart tissue has not shown any reaction between our antibodies directed against carbohydrates and structures on heart tissue. So a wild goose chase, but we opened up a whole new field of peptide carbohydrate mimicry and showed that we could transpose one class of molecules for another. And today, we do high throughput screening, and we don't care what the nature of the molecule is. It's how the receptor interacts with that molecule functionally. So functional mimicry is what we establish. And finally, accident. I'll talk a little bit about influenza A. This is like bringing coals to Newcastle, coming to the Institute of Glycomics. But as you know, there's considerable interest in designing new therapeutics. This shows the air traffic in a day across the world, and you can imagine the pandemic risk. This slide is probably familiar to all of you, but after the influenza virus attacks a host cell, it has to break away or bud from that cell, but it's attached to that cell through a certain receptor. That has to be cleaved by an enzyme called neuraminidase, and a strategy to prevent further replication is to inhibit that cleavage. Mark von Itzstein has been very active in the generation of one of those candidates that act as inhibitors here to prevent release of the virus particle, and he has generated Relenza. At the same time, other groups generated oseltamivir. And so once again, by design, we decided to synthesize a hybrid, which brings the two elements together, Relenza and oseltamivir. Mark helped us with some of the initial work here, and it turned out that this particular molecule was extremely active and active against many of the mutant strains that came subsequently. But that's not what I want to talk to you about. We were trying to optimize that lead and to generate this particular derivative where you see we have a sulfur atom here instead of a nitrogen. And instead of getting this molecule, we obtained this very unusual structure, two molecules, these spirolactams for those chemists in the audience, and these turned out to be very active against viral strains. So completely by accident here, the reaction was giving us two new compounds which turned out to be very active. So having all this going in the laboratory, why change to administration, right? And this pretty well summarizes my feeling. One of my heroes, Mahatma Gandhi, it's important to do something, you know, instead of sitting around complaining about things. And so one admits that pattern recognition, learning from one area, can be applied to another area. So I reasoned that what we'd learned in the chemistry area and the biology area could be extended to administration. I also realized that the interdisciplinary approach was the way to go about it, and not to protect one's turf and protect different fiefdoms, but to break down the walls. And so that was the model, knowing full well that an event here has an effect over there, and vice versa, and that we should not act in isolation, and we should not work in isolation, and that we should think very carefully about the different sectors and the communication between those sectors. My first challenge was to set up and change the Department of Chemistry, and I became chair of that department, and I decided to throw away all those traditional boundaries and to have only three streams: chemical structural biology, material science, and chemistry related to nuclear science. And we redesigned the department, and we were able to hire individuals who straddled one or more disciplines, who would have fallen through the cracks. And this turned out to be a very effective strategy. We boasted a little bit about it by leading the national conference, as you see here, but it wasn't enough, because we had to prove to the international audience that we were players. And so some of you were present at the International Carbohydrate Symposium, which we also ran, to show that we had accomplished in very short order an expertise in glycochemistry and glycobiology, as you have done very expertly in this institute. The strategy then was expanded as I became vice president of research. And I took on that role because I really believed that in order to take a very young university, Simon Fraser University, like Griffith, to a certain level of respect and impact, it would take a special strategy similar to what we'd done with the chemistry department. So I summarize the approach simply here: to take risks, to be agile, to be edgy in a good sense, to invest in bold research initiatives, to recruit outstanding faculty and students. That was very important. But to dare to experiment, right? To take risk and dare to experiment so that you could discover, you had the ability to discover new things, and to provide Canadian leadership. One had to build on a strong foundation, but in terms of interdisciplinarity, to bring the diverse disciplines together in this kind of a model was the mandate. And with that, we defined strategic research themes which brought together faculty from the humanities all the way to the applied sciences to work in teams. So one of the areas that we carved out, which is unique, is that of cultural evolution. And that is now, has opened up a whole new field as one merges social sciences with sciences. Has this worked? Well, let me first say that it's important to fund these initiatives. So we became shameless real estate agents and land developers. And we took some of our forest area, it's very similar to your Nathan campus, and built a city, which we called Universe City. This is like Frank Sinatra with a C. And we sold these condos, and all the revenue from those condos went in this fund. And the interest came to me, which I thought was right, you know. And we used that then to fund those interdisciplinary initiatives. And did it work? Absolutely it worked, you know. It's just a few statistics, but that's not important. Let's look at the international rankings, right? Of universities under 50 years of age, we're number 2 in Canada. Total, number 12 in Canada. The 2015 QS rankings just came out. SFU is now number 11 in Canada, right? We too are wrestling with an architecture that you are wrestling with, the Gang of 8, the Slighted 6, and the rest. Well, we have the U15, the others, and the rest. And so we share these issues. And so this was very important to show that one could derive a strategy and arrive at number 11 in Canada without a medical school, right? So the strategies worked. Total research income, of course, climbed. That generally happens. Dollars follow good ideas, right? And in terms of success rates in granting agencies, you see that for our established researchers renewing, right, in 2014, a 97% success rate, compared to an average success rate nationally of 80. For our early career researchers, 83% versus 66, and so on. So we've clearly done something. I'd like to think that it was because of strategy. Right? This was one of the highlights. One of my last initiatives was to open an incubator. I used to run student and faculty incubators and accelerators, and this is in the Bombay Stock Exchange. So if you visit Mumbai, visit the Bombay Stock Exchange, we have the 18th and 17th floor, together with Ryerson University, another up-and-coming university. This is the Governor General, David Johnston, opening our incubator with the traditional coconut smashing ceremony. Yeah. Why? Because you can, you know, and there I am, a safe distance behind. So this is an example of working with community immersed in the Bombay Stock Exchange with access to capital from the Mumbai Angels. And so we fund these student companies. At the moment, we have 60 student companies there. But it was time to move on. But this is Simon Fraser University today. It's 50 years old, 3 campuses, okay? And it's come of age. And I'd like to think that those efforts had something to do with it. Yeah? So where does one go from there? Well, let me say a little bit about advocacy. Right? I always believe this: if you want advice, you ask for money. If you want money, you ask for advice. And that's been my modus operandi with government throughout my career. And I've learned a lot. First of all, know your facts. Because when you go in to see government officials, they have sheets on you, they know more about you than your mother usually, right? And put your perspective in the context of the entire system. Do not present a myopic view, right? It doesn't help you. This is very easy to do. You let passion get in the way of facts. Well, you have to control that. And don't generalize based on circumstances in one sector. And most importantly, present a coherent view. Sing from the same song sheet whenever possible. So those are some of the lessons I've learned, right? Has that resulted in an investment? I think so. The government of Canada invested $2 billion in 2014 for research and innovation. Okay. Not a bad investment. So my next opportunity was to become president of the Natural Sciences and Engineering Research Council, to do something for Canada. Because Canada was at the cusp. The investment I showed you in the earlier slide was good, but it wasn't enough. If you talk about sustained impact, right, on a global stage. We currently have a budget of $1.1 billion annually. It's not enough. Okay? We fund 12,000 professors, 30,500 students and postdoctoral fellows. So we provide the scholarships for these students and postdocs. And importantly, we work with a stable cohort of 3,500 Canadian companies. So these companies work directly with the professors. And we bring this dynamic together at the outset so that professors work in an informed way. My motto in my incubators was kill it quickly. Right? Do the critical experiments, don't waste time, and you do that only if you have an informed view from industry. Right? And our vision is to make Canada a country of discoverers and innovators for the benefit of all Canadians. So this is Jane Goodall's observation of a chimp for the first time using a tool. So is that innovation? Well, that's social innovation, because that led to a whole different type of behavior. These are two chimps that my dad worked with when I was a boy. I used to walk around with these jokers, right? And I asked the question when I looked at this picture, sitting on a bench waiting for the bus, is this innovation or imitation? Right? Because they were sitting like men and they'd walk erect like men. Right? So you have to ask, was that innovation or were they just imitating us? And several countries have been accused of imitation and not innovation. We're followers in Canada generally, we're not leaders. And so this is a big challenge. How do we address that? Well, the first thing is you build on a strong foundation of discovery. Right? Without foundational science, it's highly unlikely that you will become innovators. But one has to admit that this is a necessary but not sufficient condition for innovation. It takes a lot more. It's a very complex dance. And so discovery, as I've showed you on the first slide, either through conception, misconception, or accident, leads in some cases to invention. But to take invention onto innovation is very difficult. And there are very few inventions that wind up on the innovation landscape. So let me switch this around and use government speak. And this is what I present to ministers. Right? What we do is we create opportunities to discovery which occasionally lead to inventions. It's important not to exaggerate. But what we're doing here in our partnerships with industry is adding value to the inventions. And the reason we do that is to de-risk for future investment. And that's the road to innovation. So it's the shared risk model. But in order to do real innovation, this is a Mobius strip, which is another one of my symbols. And I use it on purpose because it has no beginning and no end. Right? And it's important to know that it has no beginning and no end. Importantly, the Mobius strip also has one side. And so we all have to be on the same side. But in order to do true innovation, you need to do market validation. You need to know about penetration of global markets, right? Market expansion. And it's not enough to just do concentrate on ideas, research and development, and concept development. We do this very well, but it takes everything else in order to do true innovation. And I have a very hard definition of innovation. It's not innovation until it's monetized or it has societal impact. So let me finish by saying a little bit to the students in the audience. Right? You are the lifeblood of research and innovation, and don't let anyone else convince you otherwise. Right? And I was very impressioned the night before I left Paris with Sir Derek Barton. He told me this. He knew I was going on to a career as an academic. And he said, if you succeed in transferring all of your knowledge to your students, you will have failed. Now, at first I thought he was trying to tell me I didn't know a bloody thing, but what he was trying to tell me, that there's an evolution of ideas and you should learn from your students. And that you should seek input from different quadrants to generate something beautiful. As this Japanese artist. This is a collection of cutlery, spoons, knives, and forks. And you see, with the proper illumination, you generate something wonderful, right? So this is the model, right? You receive input from different quarters. But where does it begin? Does it begin at your level, grade 11? I don't think so. I think it starts when you're about that high. So with that in mind, I started this campaign with little kids, and I'd accost them in malls with my postcards with experiments, right? Giving them experiments, or handing them to the parents, of course. You know, the art of experimentation, observation, conclusion, right? In both languages, since we're a bilingual country. And this is very important to generate that curiosity in children, right? And it has to start at that early age, right? It's important to go on the road and do shows, and here I am doing one of these Stinks and Bang shows, right? With children, because it's very important to show them the power of science. And we've continued this, of course, at different levels. It's important to document one's own advances, right? We're very quick to celebrate the accomplishments of others, but we often forget about our own. So with that in mind, we started promoting Canadian chemistry. And in the old days, we used print. Now we use YouTube. Right? But the strategy, as far as students is concerned, is similar to what I've talked about. Don't be afraid to take risks, to be agile, to be edgy, to invest in bold initiatives, and to dare to experiment. And it's important in your future. It's a very different world, you know. You have to be a first mover and not a follower. And I think that's very important. And you have to know when to pivot, right? If you've been trained well and you're trained in the art of critical thought, you have to know when an idea is done, but you also have to recognize that you've probably built up this incredible team. And so you have to know when to pivot with that team and apply yourself to another situation. This is particularly important when you're starting up companies and so on. But the most important thing is passion. If you're not passionate about something, don't do it. So define your passion and follow it. This is much more difficult than you think. But I like to quote one of my favorite Formula One drivers, Sterling Moss, who used to say, if everything is under control, you're just not going fast enough. And that speaks to taking some risk. But I think this is going to serve you well. And in terms of future citizens, you know, I always presented chemistry as a core discipline, but really science is the core, right? But what we're trying to do is generate students who are adaptable to new situations. This is going to be critical, who can recognize patterns, who know how to operate in different cultural, ethnic situations in a pluralistic society. We're looking for global citizenship, and that's going to be critical. So building networks internationally, I think, is going to be the future. Ambassadors with appreciation of different countries and so on. So that's what we're striving for. In terms of PhDs, we have to return to the concept of PhD as a doctor of philosophy, not a doctor of immunology or chemistry or whatever, right? It's to be skilled in the art of creative and critical thought and analytical thought, and that combination and the ability to adapt to new situations and new fields, eventually for cultural, economic, and societal impact. Many of the jobs you see today will not exist in 20 years. That's the prediction. So you have to be adaptable. You have to be ready. And the investment in the next generation is going to be critical. So we're doing a lot of that. The objective is to land on your feet. Very simple, right? So you'll come across many dead horses. Don't flog them. Know when to pivot, right? Take a friend along in a collaborative approach, right? Hopefully you won't lose the friend along the way, right? Try and make sure you're intact. But remember that perspective is important. Others have very different perspectives. And Rabindranath Tagore said this, the fireflies twinkling among leaves make the stars wonder. And it's very important to recognize that all of us have different perspectives. We bring different things to the table. I'm not claiming that it all works out, but sometimes it does work out, you know, and you land the big one. So it's a journey, and sometimes it does work out. Let me finish by showing you some pictures of my various groups. I've been many, but you'll see a very international group, and it's very important. Some of these people had never eaten anything but meat and potatoes when they joined me, but when they left, they knew all about curries, and they knew all about all religions, et cetera, et cetera, et cetera. So different nationalities, Ecuador, Cuba, Sweden, right? China, the UK, China again, India, Germany, Chile, China. So it's important that social interaction was very important. I had many parties in my place, and this shows one of them. You'll see again people that learn from each other and learn about ethnicity, culture, religion, etc. It's important to celebrate, and I dabble in music, so we celebrated at every turn, right? And you'll see some of these early shots, a little bit more hair. That's a band I had in graduate school, but we've continued to celebrate at every occasion. So every paper, every grant, or every scholarship, celebrate. It's important. Right? But this is the bottom line. Training PhD students in the art of critical and creative and analytical thought. That's it. With the sense of international purpose, with clarity of vision about what the future of science and technology is going to do for mankind and womankind, and how building that network is going to play a role in the future. I noticed a lot of high school students here, so I inserted this slide at the last minute. This is my high school student. I was giving a public lecture, and he came up to me and he said, I want to work for you. And I looked at this kid and I said, are you sure, you know? And he said, oh, yeah. So I said, come and see me. And he came to see me, and I realized right away, he was in grade 10, right? He was 14. And I realized right away that I was dealing with a special person. I said, all right, well, let's start you off in the lab. And I started him off as a computational biologist, and he wrote some amazing algorithms, but he was very bright. He understood why he was doing it. So while he completed his 2 years of high school, grade 11 and 12, he also completed years 1 and 2 of a science degree at Simon Fraser University. He got an A+ in every course. He had women around him he didn't know what to do with, you know. And I had to tell him that it was, I said, Eric, it's for your brain. It's for your brain. He said, just leave me alone, right? So, but he was an all-round person. He was a BC gymnast. Parallel bars was his event. He played saxophone in a jazz band, and he was a very good cook. And so every group meeting, he would bake pies for us, and Yoon can vouch for that, you know. So he finished his degree. We got 3 publications together, right? He didn't finish his degree, he finished 2 years. And he went to Oxford last year. He completed year 1, and he came second out of 99 students. He's captain of the gymnastics team. His shoulders are out to there now. His rings are his event. And he captained the Oxford gymnastics team against Cambridge. His brother is on the Cambridge gymnastics team. So, you know, it's just a pleasure to work with students who have that kind of passion. He's going to be a Canadian leader, I guarantee it, when he comes back. So I'll finish with that. I'll thank the granting agencies who funded all of our work over the many years. NSERC, and I'm now with the president, Curious, and the Canadian Institutes for Health Research. And I'm very grateful to these people because they make it come alive. So with that, ladies and gentlemen, I thank you very much for your attention. Yeah.